Assalamualaikum and hi, my name is Warni Izzati binti Wakalik. The number metric is S59129. Today I'm going to present about species identification of non-hybrid and hybrid Pangasiid catfish using polymerase chain reaction. First, we will go to the introduction. Identification of Pangasiid species based on morphology can be effectively applied in adults. However, the morphological identification guidelines for larvae have not been well established for all Pangasiid species. The Pangasiid species used in this research are in point number 2. Among the hybrid species, they were capable to reproduce, and this will increase the risk of genetic introgression among species, as has been shown in some fish species. Moreover, the hybrids could not be unambiguously differentiated based on solely morphological characters, and this may lead to an admixture of parental species with hybrids, leading to the genetic contamination of broodstock. Furthermore, morphology-based species identification is not possible when the processed product of such fish are examined. So, the problem statement from this research study is the issue of incorrect species for food supply in food industry and in verifying the species origins from various forms of product. The objective of this research study is to be able to differentiate between hybrid and non-hybrid fish Pangasiid species using polymerase chain reaction. And the second objective is to identify 6 economically important Pangasiid catfishes using PCR-RFLP markers. Next, we will move on to the methodology. First, fish samples. Wild or captive adult and post-hatch larvae of Pangasiid species were collected from various locations in Thailand. The fin clips of approximately 1 cm were dissected out from the adult fish and placed in obsolete ethanol. The whole bodies of post-hatch larvae were collected and preserved in obsolete ethanol. Specimens were transported back to the fish genetic laboratory at Kasartrat University Bangkok, Thailand, and kept in -20 degrees Celsius until needed. The second step is the DNA extraction. Genomic DNA was extracted from each specimen following the standard phenylchloroform protocol. Concentration of extracted DNA were spectrophotometrically determined. Genomic DNA was kept at 4 degrees Celsius until used. The third step is the bioinformatic analysis. The genetic diversity and population structure of 6 Pangasiid species has been previously published based on the polymorphism of 16 sequence rDNA. Then, we will do the restriction analysis of mitochondrial 16 sequence rDNA. And then we will proceed with the restriction analysis of nuclear LGMH. Restriction pattern generated from each restriction endonucleus were given letter designation alphabetically according to their appearance. The composite PCR-RFLP patterns were constructed from combinations of restriction patterns of 16 sequence rDNA and LGMH. And for the last steps is this ethics statement. All these experiment procedures using the animal conform to the guidelines established by the Animal Care Committee, Kasartrat University, Thailand. The table on the screen shows the result. As for discussion, species-specific PCR-RFLP markers were successfully developed for reliable species identification of 6 Pangasiid species consisting of Pangasianodon gigas, Pangasianodon hypothalamus, Pangasius buckorti, Pangasius consophilus, Pangasius leronaudi, and Pangasius sanimonse adults, and also larvae by using restriction analysis of 16 sequence of rDNA with a minimum of 3 restriction enzyme. These molecular genetic markers have facilitated the correct species identification of adults and early development stages egg or larvae of fish, which is useful for fishery biology studies. PCR RFLP markers have been used to identify adults and egg or larvae for stock assessment and indicating catch rates. So, the species diagnostic PCR RFLP patterns can be utilized to identify seed species and their distribution in natural sources. Due to increasing demand for Pangasianodon hypothalamus in global markets, the authentication of Pangasiid catfish and catfish-derived product is important. PCR RFLP results demonstrated that the markers had more advantages than the traditional DNA barcoding. So, this means these markers are useful for authentication of Pangasiid catfish and catfish-derived products in the food industry. Interspecific hybrids may pose a serious threat to native species. Misidentification of parental species and hybrids may easily occur if the identification is based solely on morphology. PCR RFLP has been used as an efficient tool for the detection of hybrids in aquaculture and of genetic introgression in wild stock. Furthermore, the molecular markers developed herein will facilitate precise monitoring of genetic introgression in natural populations and hence may lead to the development of conservation plans. This is the reference that I use for the individual assignment. Thank you.